Muy buenas tardes, les damos la más cordial bienvenida a esta segunda conferencia en este bloque sobre terapia, sobre genética y genómica médica y humana. Soy Gabriela Repeto de la Facultad de Medicina Clínica Alemana, Universidad del Desarrollo en Santiago, Chile, y tengo el tremendo honor de presentar a la doctora Nara Sobreira. Voy a compartir aquí una lámina. La doctora Sobreira es profesora asistente del Departamento de Genética de la Universidad de Johns Hopkins. Ella estudió medicina en la Universidad de Pernambuco y realizó una doble formación como genetista clínico en la Universidad Federal de Sao Paulo y en la Universidad de Johns Hopkins. Y tiene un doctorado también en genética humana en la Universidad de Johns Hopkins. Ella ha trabajado por mucho tiempo en enfermedades raras, en aspectos genómicos y también en aspectos relacionados con data sharing y también en descubrir nuevos genes de enfermedades y así como comprender su fisiopatología para avanzar en terapias. Ella ha sido una de las co-creadoras de PhenoDB, que es una base de datos fenotípica y genómica abierta. Creó el PhenoDB Variant Analysis Tool que muchos de nosotros utilizamos y es una de las co-creadoras de GeneMatcher y VariantMatcher y de Matchmaker Exchange, que son herramientas muy útiles para compartir información de Next Generation Sequencing para el beneficio de los pacientes, de los clínicos y también de los investigadores. Les invitamos a todos y todas los que nos están escuchando a enviar sus preguntas por escrito en el chat. La doctora Sobreira hablará en inglés, y pueden mandar las preguntas en castellano, inglés o portugués, no las arreglaremos. So Nara, great to welcome you to our ALAG meeting, and the screen is yours. Thank you.
Thank you so much, Gabriela. Thank you for having me here to talk to you today. Such a great meeting and after such a great talk. So let me share my screen. And like Gabriela said, today I'm going to be talking to you about data sharing for the identification of novel disease genes and variants. More specifically, a little bit more details about GeneMatcher and VariantMatcher data tools that we created here at Hopkins as part of a sequencing project that we had going on for 10 years that was called the Baylor Hopkins Center for Mendelian Genomics. So we all know that in the last 15 years, next generation sequencing created a huge amount of data. And this amount of data that was created facilitated and is facilitating many disease gene discoveries. And we just heard an amazing talk about how discovering a gene responsible for a rare disease can help with treatment. It's a long way, but discovering the gene can be the start of something amazing like what we just heard in the last hour. So it has helped, and many gene discoveries have been made because of the advancements in sequencing. And here I have a chart that comes from data from OMIN, a database that catalogs the rare diseases and the genes that cause these rare diseases. And you can see year by year the increasing number of genes on the orange bars and phenotypes on purple bars. And you can see how this progress was amazing in the last 15 years and mostly due to the use of next generation sequencing. It really kept us going with gene discoveries for rare diseases. And the numbers went up from the number of genes associated with phenotypes listed in OMIN went up from 2,500, that was 12% of our genes, if we consider 20,000, the number of genes in our genome, to 4,500, that is 22% in the last 11 years. So it has really helped But many other things beyond just generating the data have helped to actually discover the genes. And one of the things that have made these discoveries possible was the data sharing. Very early, we came up with well, we all knew from old days that when we had a candidate gene, we needed to go out and find other people and talk about our findings to see if we could find other patients and other patients with mutation in the same gene. So very early with the amount of data that was being created, people came up with ideas that they needed to share the data. This data needed to be shared. It could not be kept with one researcher or a few researchers. If we really wanted to take as much advantage of this data as possible, we needed to share the data. And as of today, we are still working on ways to share the data in a way that is useful and can lead to more and more discoveries. So data sharing is an important piece of creating this data and going from creating this data to finding important things, meaningful things in the data. But it's not an easy task. From early on, many questions came up when we started thinking of how to share our data and talking to other people who were creating data, and many questions came about, and questions that are still being discussed nowadays have not been completely answered. And to be honest, we don't have a perfect data sharing tool. Each one of them has its strengths and its weakness, but we are still working, trying to address all these problems. But some of the questions that we had and that we have when we are creating a database to share data, genomic or phenotypic data, what data is needed for the discoveries, what kind of discoveries we want to do, and what data is needed to be shared to actually lead to these discoveries. The discovery for disease gene or discovery for variant, a causative variant. And what data can be shared, but still ensuring that the patient confidentiality and privacy are respected. So it's not just go around sharing any and all data. We have to ensure that the patients are still safe and that the confidentiality and privacy of the patients are respected. So how to share the data and how to share the data in a way that makes sense, where and to whom, right? So everybody should get all the data. Who should get what data? And IRB has helped many cases to solve these questions, or at least to guide us through how to share the data in a way that we are still protecting the individuals the data come from. But it's a thing that it's another step that we have to think of when we are creating a data sharing tool. It's like what the IRB requirements are on our center, on the centers where this data was created. And one thing that has been from the beginning a very interesting piece to deal with was how to ensure that the researchers that generated the data are recognized for their work. Once you share and someone has a finding based on that data that you created and you shared, how to make sure that you are recognized. That was a problem from the beginning, because now 10 years later, we see a huge change, and it's actually very good to see and makes us happy to see people sharing without so many reservations. But 10 years ago, people really didn't want to share the data because they were really worried that they would be scooped, that someone would get the information that he was sharing and write a paper or publish without including them on the publication. And unfortunately, that's not just because people want to appear and to be important. Our system requires us to publish to get more funding to do more research. So once you don't have publications, you kind of get behind on the ability to generate this funding and continue doing your research. So we have to think that on a way that we are fair to everybody when we are sharing data. And I think everybody keeps that in mind when these databases are being created. So in the last 10 years, many databases have been created in many different countries, and I'm not going to try to list all of them here. I probably don't know of all of them. What I'm listing here is what I know of and what I use the most. So it's far from being a complete list of data sharing tools or databases. We have different levels of data sharing. So for example, here in US, there are some NIH-funded databases, and NIH has more and more requested that the projects that are funded by them to agree to put the data, to share the data through these databases. These databases are not public. You have to actually go through a process to become a registered researcher with access to the data, so they can ensure that the data is being used in a compliant way with the IRB requirements from NIH and from the institution where this research is coming from. But some of them, the one that's probably the oldest and very well known is the dbGaP. So a lot of genomic data and phenotypic data from projects that have been funded by NIH since the time of GWAS are here. And then more recently, very recently, in the last, Cavarica is probably in the last 5 years, and EnView is in the last 2 years. These two new databases are also connected to dbGaP, but places where researchers funded by NIH for specific projects are actually putting that data there, the genomic data and the phenotypic data. Another thing that has been changed a little bit is more and more the researchers have realized that the genomic data needs the phenotypic data aggregated to actually to be able to make sense of the full genomic variants that we find. So EnView is the NAGRI, it's a branch from NIH, Genomic Data Science, Analysis, Visualization, and Informatics Lab Space. In these places, you can also, in EnView and Cavarica, you can also do many different kinds of analysis on your data. So it's not only a place for you to go and download the data that you want and work on your own lab. It's a place where you, in there, you can do analysis also. The same Cavarica that was created in aggregated to the Gabriela Miller Kids First project. That's a genome sequencing project for congenital anomalies and pediatric cancer. So now NIH is actually working a way to connect these databases. So we don't have the data from different projects in different databases, and we have to be going to 3 different databases to actually get our data or data from some other project that we want to work on. They are working on connect them so you can go to one place and have access to all the data that is in different databases from NIH. But this is not publicly, as I said, is restricted. We do have many public databases that share many different kinds of data, different levels of data, and also sometimes with different access to different level of data. So when I put public here, I meant mostly you don't have to pay for. Some of them you don't have to, that they don't require any registration or anything. You just go there and you make your query and you get your answers. But some of them you have to create an account, be registered, be approved to be able to get the data that you want. Some of them have the 2 levels. Some data you can get without any registration required, and some of the data you actually have to register, be approved to have access to the data. Some of them also have phenotype data available. So the UK Biobank, it's one that is very well known lately. They have thousands of sequencing that was done in many individuals. Some are healthy, some have different diseases, and they have phenotype. They have, I think, very, very detailed phenotype, as detailed as possible, phenotype information in there. This is one that you actually have to create an account and be accepted be approved to be accessing the data and doing different kind of use of the data in there. This cipher that started as a database for copy number variation, also with very significant amount of phenotypic information associated with each of the copy number variations that is listed there. But they also have now single nucleotide variants. In the last years, they have included also single nucleotide variants to the database, not only the copy number variation. It's an amazing database to work with. Also, some data are publicly available without any registration required. Some data you need to actually register to have access to. There are newer ones from Decipher. UK is pretty new, but like Decipher is, I think, is the oldest one in this list there. And you have newer ones like Genome2MP that came out also from the CMGs, the Center for Mendelian Genomics. I told you we were part of the Baylor Hopkins Center for Mendelian Genomics. So there was another center that was in UW, and they created Gene2MP to share the data that they created as part of their project. They also have phenotypic information associated with variant level information. And ClinVar, that most of you, oh, I don't know most of you, but many of you probably know, that is a database that's NIH funded that was created to aggregate the variants that clinical labs were reporting as benign or pathogenic. And they went on to also add variants that are published, variants that are curated by OMIM, and even researchers, when you are writing a paper or you have defined that that variant that you are looking at, it's the variant that you want, you can submit your variant there. And actually most of the papers, the journals in nowadays, when you are going to publish a case and you're describing a gene, a novel disease gene or novel disease variant, they require proof that you have submitted that variant to a data sharing database. And I think the one that people go the most is the ClinVar one. And then some of them don't have phenotype data, but they are very important for our analysis, right? When you are doing analysis of rare disease and trying to find a causative variant, the first thing you want to say is like, this variant is rare. And many of these database, I put here, I think in the order, like the exome variant server was the one that we used with 6,000 exomes at the beginning. And then we had the 1,000 genomes project. And then we have the EXAC with 20-something thousand exomes. And now we have NOMAD with, I think, about 150,000 individuals, most of them exomes, but I think more than 30,000 genomes. Or actually, I think the last one, they had 70,000 genomes that were coming out. So now we are talking about 150,000 individuals with exome and genomes in NOMAD. So it means we have a much better database that's publicly available to infer if the variant is rare or not. The one problem that we still have is the population that you are studying. And it's very important for us in South America. I'm from Brazil. Many of the patients that I sequence are from Brazil. And when I find a variant that it's rare because it's not in NOMAD, the first thing that comes to my mind is that variant rare because it's really likely to be pathogenic, or if this variant rare because it's from a different population that's not represented in NOMAD. Many populations are not represented in NOMAD, so we have always to have that in consideration. I have Human Gene Mutation Database is another variant level database that tries to keep the variants that are published in there. So if you want to see if a variant has ever been published before and how it was classified, you can use Human Gene Mutation Database. And the last one here, it's Abraon. I don't know if any of you have heard about it, but Abraon is actually a Brazilian database. And it has the genome sequencing data from 1,171 individuals from Sao Paulo. It's a very important one for us. Like I was saying, it's more representative of our own population. And every time I'm studying a Brazilian patient, I find a variant, I now go to Abraon on top of looking at NOMAD to see if the variant is really rare. Of course, 1,171 individuals from Sao Paulo is not representative of the entire Brazilian population. It gives me a better idea, but it is still very far from being representative of our mixture in Brazil, right? So this is important. It's important for the other countries in South America because many of them would not even be represented in a large sample from Brazil. So we need to have that in mind, and we have to actually try to get our own population represented in these public databases to improve the research of our own population. So with all these databases, what do we still need? As I said, neither one of them is perfect. We have different kind of database assessing different needs, and we had our own need, and we thought of what we wanted to, what was missing for us. For us, studying rare Mendelian disease, one thing that was important that was missing was detailed phenotypic features. Like we need, when we find a variant in a rare patient and we find a candidate gene, we need to compare detailed phenotypic information to be sure that we're talking about the same disease. Family history, very important for us also as geneticists dealing with rare Mendelian diseases. And because sharing this kind of information in a safe way is not that easy, one thing that was missing was, okay, maybe I cannot have that in the database available publicly to be accessed by anybody, but if I can at least reassess the individual, if I can go back to the person who deposited the data in there and talk to this person about this individual and get the person to maybe go back to the person that is the patient that had that variant and investigate more based on the patient that I'm talking about. So these were things that we really wanted to have access to, and to fill these gaps, we were thinking of a data sharing tool that would enable us to get this data that was missing. And how would we do that? So we think that to get around IRB requirements and also to get around the concerns of researchers about data, sharing the data and being scooped, that was actually a concern that we had to deal a lot 10 years ago when we created our first data sharing tool. We said, okay, if we share the least amount of data to begin with, and we just get the connection started, but we also share contact information of both parts, then they know they have a shared interest and they know each other contacts information, and they can go on and talk to each other and exchange more details. So we decided that our first data sharing tool would share basically a gene name. I would go there, I would create an account, and when I created an entry for a specific patient that I was investigating, all I would need to put there was the name of a gene of interest for that patient. And if Gabriela did the same thing and she created an entry with the same gene, now the database would connect Gabriela and I, and we would have each other's contact information, and we would decide if we wanted to talk to each other more and exchange more information about the patients that had the mutation in that gene. And that notification would be simultaneously. Gabriela would know at the same time, I would know that we had interest in the same gene. That was the way that we thought would be the most useful to get the information that we needed. Now I can get detailed phenotypic information. Now I can get detailed family history. Now I can ask Gabriela to go back to her patient and make sure that they don't have a cardiac anomaly. So that was the way we thought it would work well. And to do that, we created GeneMatcher. So I don't know if you have heard of GeneMatcher, but basically GeneMatcher does what I just described. It's a tool to connect patients, family members, clinicians, and or researchers anywhere in the world from any country that have interest in the same gene. We also can connect based on variant level, phenotypic features, and an homeme disease ID. But PhenoDB is mostly used for gene names. Usually you did the analysis of a patient, you have a candidate list. We don't let anybody put more than 10 genes, so we can make sure that people are selecting the best candidate genes for that patient. And you say, okay, these are the best candidate genes for my patient. I'm going to create an entry with these 10 genes in there and see if anybody else in the database has an entry with the same gene, the same one of these 10 genes, or more than one. So the data is basically de-identified, right? Because the only thing you're putting in there, it's a gene name. You can put variant level information also if you want, and you can put phenotypic features in there also if you want, but all you are required to do is to put a gene name. And this is not identifying anybody. The matching is automated, as I said. If there is anybody else in the database with an entry in a gene that is the same as yours, both sides will be notified at the same time, and you choose to follow up on it. Oh, I know Gabriela, and I would love to collaborate with her on this project. Let me contact her. You know what? I don't get along with Gabriela, and I don't think it would be a good idea to me to talk to her. Let it go. Oh, I don't know such and such. And don't know Gabriela, but I want to solve the problem of my patient, and I would just send an email to her and see what happens. Can tell you that it has been very successful. I'm going to show you the numbers, and you see that it has worked very well. So these are the numbers from 2013 when we started to 2021. So we are almost 10 years. I didn't even realize that we will be 10 years in 2 years. And as of October 21st, there are 13,000, almost 14,000 genes from 12,000 submitters in GeneMatcher from 94 countries. And of these 14,000 genes, almost 9,000 of them have matched. So have generated a match. But you can see that many of these genes actually have not matched ever with anybody in the database. At the end of this talk, I'm going to show you a paper that we just published that's actually doing a more detailed analysis of these genes. What's the difference between the genes that have matched, the genes that have not matched, the gene that have matched more than 10 times? Is there any difference, and why do we think that these differences are in these happening among these 3 different groups of genes? The ones that match, the ones that don't match, the ones that match too many times. But you can see here that that recipe has worked. People are using it, and people think that it's easy to go there, create an account, put that gene of interest, and try to find other people that have interest in the same gene. The interest doesn't have to be specifically another patient with a variant in the same gene. Sometimes it's, I want to find someone who is doing research on this gene, and maybe would be able to do a functional test on my variant to see if the variant could be pathogenic or not. Or sometimes someone is working on a mouse model and is interested in the same gene, or someone works with flies and would love to do a fly model for the gene that I'm looking at. So all these kind of connections can be made through GeneMatcher. You just have to say the gene and see if anybody else is interested on the same gene that you are. As I told you, we have 94 countries, and I did focus a little bit more on the South America countries, so you can have an idea of what's happening in South America. Here, most of the countries have actually looked and used GeneMatcher before. you can see here that it's alphabetic order. So we don't have a preferred one. I didn't put Brazil on the top. So you have Argentina with 34 users. This is the number of users, it's not the number of genes. The number of genes are larger. We have 197 from Brazil. I think that's also my bias because I talk so much about it in Brazil. A lot of submitters from Chile, 44, Colombia 13, Ecuador 7, and Venezuela 1. Still have few countries that have not used it, and I'm hoping that maybe today there are researchers from these countries that have not heard of GeneMatcher before, and now will think that this is useful for them and can go there and start using it. Who are these users? Who has been using GeneMatcher? Most of the users are researchers. About 8,000 of our users do identify themselves as researchers. And you remember that we have a total of 12,000 users. So 8,000 of them recognize or describe themselves as researchers. Then we have about 5,000 of them that describe themselves as healthcare providers. And remember, one doesn't exclude the other. You can be more than one. 677 patients. And this is actually a population that I would like to increase using GeneMatcher. We have not had we really reached out to them specifically. These patients just hear about us from another clinician, from sometimes they are researchers themselves, but I would like to actually work on connecting them to GeneMatcher, specifically talking to them about it and how to use it. And then we have, I think most of the people actually participating on this meeting may not be people who work with human patients, but do research using model organisms. So who are the model organism researchers that are using GeneMatcher, and what's the amount of data from other organisms that we have in there? We don't have a lot. Of the 14,000 genes that are in GeneMatcher, 338 of them have been submitted because they were being investigated in another model organism. 203 from mouse, 55 from zebrafish, 44 from fruit fly, 21 from yeast, 15 from a variety of other non-human organisms. So this is another group of investigators and researchers that we would like to increase in GeneMatcher, using GeneMatcher, and we are doing some things towards that, and I'm going to tell you about that in a little bit, and also maybe tell you about database that will be very useful for you. How has GeneMatcher helped? I'm sorry, the sun is kind of getting on our way, but how has GeneMatcher helped? This is the number of publications that have cited GeneMatcher so far, and there's only one way that we can actually have an idea. It's if GeneMatcher is a knowledge on a paper, and then we can query PubMed and get this paper and put on our publications page in GeneMatcher. So in GeneMatcher, there is a publication page where you can see all the papers that have acknowledged the use of GeneMatcher throughout their work. And we did an analysis of some of these papers, and we had a date of like June 12, 2020. And at that time, we had GeneMatcher knowledge in 325 publications. So you can see that in a little bit more than a year, it has been more than 200 papers that have been published. So it's actually increasing the number of papers that are being cited by GeneMatcher per year. But when we had these 325 publications in 2020, 302 of them, they described actually 302 genes in 249 publications. So many of the publications didn't describe a gene specifically, but 249 of them did, and they mentioned 302 genes. Of these 302 genes, 157 were related to an autosomal dominant phenotype, 105 related to an autosomal recessive phenotype, and 24 were related to autosomal dominant and autosomal recessive phenotype, and 6 were related to an X-linked recessive or dominant phenotype. So it's still solving the autosomal dominant diseases looks like it's a little easier if you're looking through the GeneMatcher publications, followed by autosomal recessive. I really think we are not doing a good job on sequencing and analyzing the variants on chromosome X. I think we could be doing that better with the sequencing methodology itself. Maybe now that we're using genomes more than exomes, that will help the discoveries on chromosome X. And this is what we got from this analysis at the time with these 302 genes that were acknowledged in these papers. Then we still had, like, of these 302 genes, 221 of them, 73% of them, were on GeneMatcher for more than a year before the publication. So it correlates a little bit with what I just told you, that looks like we are getting more publications per year in the later years. And this is because once you find your match and you say, okay, this is looking like a good gene, there are other steps that you will take before you write a paper. Sometimes you want to do functional studies based on cell line work. Sometimes you want to do mouse model work or other model organism work, and that will take time to actually go from that match to the publication. 231 of these genes are now actually associated with a phenotype in OMIM. So they have been published, and they actually have been recognized by OMIM as a disease gene. And 73 of them already had the OMIM phenotype before they had been posted in GeneMatcher. We usually say that GeneMatcher is better used if you put their gene that has not been associated with a disease before, to be a completely novel disease gene. But we also found that sometimes a gene can be causing more than one disease. And sometimes the phenotype of your patient may be slightly different, and that can uncover a phenotypic expansion. So many people do use GeneMatcher if they find a variant in a gene that's known to cause a disease, but the phenotype of their patient is slightly different or completely different. And we can see here that 73 of them were actually published, even after they were in OMIM already. Then we get to matchmaker exchange. When we created GeneMatcher in 2013, there were another group creating a similar database that was from Canada, and it was Phenome Central. They were creating a database, and then they wanted to match based on phenotype. They didn't want to match based on gene. We thought matching on phenotype would be very complicated. Dr. Hammer is a clinician, I'm a clinician, we know how hard it is to actually well phenotype a patient and match these phenotypes. We thought that it would be very hard to do through the computer, but matching on a gene name would be much easier. So they started with phenotype, and we started with the genes. Now we both do both. But we decided that it would make sense that we were connected. So whoever wants to use GeneMatcher doesn't have to go to Phenome Center and create an account and put that data all over again in another database, and vice versa. They can just go to GeneMatcher, and from there, click on a button and send the query to Phenome Central also. And if there is a match in Phenome Central, they will be notified of it. So Phenome Central and GeneMatcher got together and started the creation of this API that would connect these two databases. And from there, Matchmaker Exchange was created, and other databases were created with similar function, but they were in different places, in different countries, and they could not just put that data in one database or the other. They had their own IRB requirements, So it would make more sense for them to have their own database. So we have VariantMatcher from Europe. Decipher was also early. As soon as we were connecting, Decipher joined us to connect, also from UK. Seeker is not an open database. It's based on Broad, but it's only for the data that comes from sequencing done at Broad. But once the sequencing is done there and they have the candidate genes, they push it out to match in these other nodes. And there is the IRUD that's from Japan, RD Connect also from Europe, MyGene2 from UW, and Phenome Center, as I told you, from Canada. So these are the nodes that are connected as of today, and few others are actually joining soon. And I'll talk to you about one of them in a few minutes. That's the model organism one that I told you that maybe you would like to look into it. But this is the Matchmaker Exchange page, and you can go there and find out more about the nodes that are connected through the Matchmaker Exchange API. And these are the ones that I just told you, and the number of patients that each one of these databases have, and the number of patients that each one of these databases have sent through the API to the other databases that are connected. And here, the number of genes in each one of these databases. So you can take a look and you can learn more about each one of these databases, and you can decide which one you want to use. Maybe you are not sure you want to use GeneMatcher. Go there and find out about the others and decide if there is one that it's a better fit for the data that you have. Now comes to the database that I was telling you. So there are other databases that are now looking into joining the Matchmaker Exchange. We have ModelMatcher, that's the one that I'm very excited about. They actually did a database for us to identify model organism researchers. So you can go there, create your account, and tell a little bit about you, and put the genes that you are interested on and what organism you're working on. And then you can put your information there to be queried, but you also can query the database looking for other researchers that have interest or that are working on the gene that you are interested on. So they are now connecting through the Matchmaker Exchange API, and soon when you put your gene in GeneMatcher, you can also send your gene to ModelMatcher and find who are the researchers working on mouse model for that gene or on a fruit fly for this gene. So I'm very excited about it. We also are looking into this other database that's called Paradigm. It's from Europe, and it does a similar kind of work. You put your information in here. You can put a gene symbol. You can put actually an affiliation of a researcher, a country, goal terms, and it will tell you who are the researchers that are looking into that gene that you are interested on and what kind of model organisms they are looking at. So I think that's very useful. More and more we want to test. It's an important piece of us defining that this gene is causative of that disease and that variant in that gene is causative of that disease, to actually test what is the phenotype of a model organism with that variant. And it's very important for us to be able to find researchers around the world that can do this work. To me, doing that is very, very exciting because, like, I remember when I was a resident in Brazil, trying to make the diagnosis of the patients that I saw in clinic and only have access to a karyotype, or actually fighting for a karyotype to do the investigation of the patient. And now having all these tools that allow me to connect to these people in any institution around the world, I think this is a big step, and it has been very fast, I think, in the last 10 years, how we became much more able to access people anywhere in the world to talk about our interests with them. And I think that's amazing. So now I go to the last part, and it will not be as long as the first part, but this is our next two. It's VariantMatcher. What I was telling you was gene level data sharing, right? Most of these databases that I just told you, they work based on a gene name. You put your gene of interest and you find other people that are interested in that gene. But what if you want to find other people that are interested in the same variant that you are? How do you do that? When you go to Nomad, you put your variant in there and it tells you that they have 10 individuals with that variant. But this is it. You cannot access these patients. You don't know any phenotype information. You cannot contact the people who put that data in there. It's all you know is that there are 10 people in there that have that same variant. And it's a problem sometimes when you're working on a disease that is maybe not so rare, maybe it has incomplete penetrance, maybe it is a late onset. Right now, I'm just studying a case and there are 6 patients in Nomad with that variant. And I'm like, yeah, it would be great if I could reach out to this submitter and know what these patients really had to define if that variant is causing the phenotype of my patient or not, right? This is what we're trying to do with VariantMatcher. So I told you we had this project going on at Hopkins. That was the Baylor Hopkins Center for Mendelian Genomics, and it's actually going to be over now in November 31st, but we are going to continue hopefully doing sequencing work and data sharing work. We know data sharing work we will continue doing, but sequencing from other projects and continue to put data on these databases and everything. But as part of the Baylor Hopkins, we created this database, PhenoDB, that Gabriela was telling you. In our research database, that's the Baylor Hopkins Center for Mendelian Genomics database, we have almost 9,000 families that have been sequenced and that we have collected phenotypic features from them. So we have, using this database, 1,828 users from 65 countries. So we have a rich database with a lot of patients that are sequenced and that we have detailed phenotype information from. Using this database, we have discovered more than 314 novel genes. We have discovered variants in 361 known genes at least, and of them, 167 have been associated with phenotypic expansion, and 24 of them have been associated with a second different phenotype associated with this gene. The numbers are actually bigger than that now. They grow every month, but we have all this data here. We have genomic data from 7,724 individuals. And every day I get a message saying, Nara, do you have a patient with variant X? Nara, do you have a patient with variant Y or Z? So I decided to automate that so people didn't have to come through me and contact me to ask about it. And this is when we created VariantMatcher. In VariantMatcher, we have the data of these patients available to be queried on the variant level. You will come here and you will say what variant you are interested on. You have the option to put the features of your patient. And if you do so, when there is a match and the email notification goes, you will get the phenotypic information of our patients in our database. And this search is done today, and this is it. But if you want to save your search and run it every month, you have that choice also. So if we put more sequence information in the database between now and next month, when your search is redone next month, then you will get the information about this new sequencing that was uploaded in the database. Here is the data that is saved. You can save your variant with your phenotypic information and it will rerun every month. So what's available in GeneMatcher, in VariantMatcher so far? It's the rare cold invariants that not include the synonymous, single nucleotide variants, rare single nucleotide variants, coding, not including the synonymous, from 6,235 individuals. And this sums up to 896,000 unique variants. As of now, we have 695 submitters from 44 countries that have registered in VariantMatcher, and 4,400 variants have been queried, and 153 of them have had a match to 1,248 individuals in VariantMatcher. When we had 61 variants, we did a analysis of the 61 variants that have matched, and they were in 56 genes. 40 of them were known to cause a disease. The variants were all very rare, but all the matches that we got end up not being a phenotype match at this point. They had not been a phenotype match when we had the 61 variants that had matched to our database. And most of the time, we were able to actually classify that variant as benign because the matches in our database happen to many individuals with different phenotypes, sometimes some affected, some unaffected. So we were able to help to classify the variant as benign. So if the variant was a variant of uncertain significance, or you are thinking of that variant as a possible novel disease variant, you were able to rule that out because of the matches in the phenotype of the individuals that were in VariantMatcher. We always say that people have to think of incomplete penetrance, variable expressivity of the phenotype, and age of onset before defining that the variant is benign because the phenotypic match doesn't look like it's happening. We are now connected to Beacon Network. If you go to Beacon Network, you can put your variant and it will tell you do a search in many databases and tell you what these are the databases that have this variant, and you can see that you can now see the VariantMatcher connection here. And we are changing VariantMatcher so we can actually make searches that are on variant, but you can select what kind of zygosity you want. I only want to match to individuals that are homozygous for this variant, and also to match in a group of variants in a gene. So you can go now and say, it's not out yet. You will be soon. This is the test page that I got a screenshot. But you will be able to say, I want to look at this gene for variants that are single nucleotide variants or indels, in patients that are heterozygous, homozygous, or compound heterozygous, and that have feature X, Y, and Z. And then I will tell you in our database what are the patients that fit that group of features that you selected. We are now looking into connecting this database, VariantMatch, to other variant level databases. We are going to use this Data Connect API that we're part of the working group that just created that. It's a GA for GA's discovery work stream product. And we are looking to connecting VariantMatcher to other databases like Gene2MP and Franklin that are variant level databases. If you want to learn more about any of these databases that I was just telling you, PhenoDB, GeneMatcher, and VariantMatcher, you can access the paper that we just published a few months ago. And with that, I will finish. I'm sorry I didn't left a lot of the time for questions, but thank you very much.
Thank you, Nara. This is a fantastic overview of a tremendously useful tool. I think that data sharing has been like a big culture change, but we say a lot about it, but to really have the actual tools that are easy to use, I think it's really bringing this a whole new step forward. So we have just very few minutes for questions. So I wanted to ask you, you showed that people in Latin America are using it. I don't know if you have more detailed information about the success of those matches, and particularly sort of, within the Latin American community, perhaps for, we always think that there are unique variants or unique diseases here. Is this a tool that's helping bring that to discovery?
So I don't have, but I can actually, so when I was writing another paper, we're trying to find how many collaborations GeneMatcher had enabled. Mm-hmm. And it was very hard to ask to actually find through PubMed the institutions and the countries from the authors. Mm-hmm. But this is something that maybe I can try with this small number of papers, like this 512, mm-hmm. specifically see how many of them have institutions that are in South America. And then I will have a better idea. I don't have this number, Gabriela, but I will do that. So we can have an idea of like how many of these papers actually include institutions from South America. That will be very interesting to see.
Yeah, yeah. And also another question, sort of moving beyond the immediate connection and a potential paper, do you have a sense of whether these new discoveries are being now included, for example, in diagnostic panels or you know, increasing the number of diagnoses of a particular disease? I know these are sort of longer term questions, but I know that you always look at your data very thoroughly.
Yeah, so that data that number will come from the number of genes that are actually in Omine. So here,
yeah,
when we had these 302 of them, 231 were associated with phenotype in Omine. And more than 99% of the times when that happened, these genes are automatically included in a panel or available for clinical tests in a clinical lab. So that would be what I would say about 76% of the genes that are described as GeneMatcher collaboration will be now in a clinical level available in a clinical lab, I would think.
Yeah, and be useful to the next patients and clinicians.
Yes, exactly.
Researchers, yes. Great, thank you very much. So I'm sorry that we ran out of time. We could think about like a longer workshop or something to help us become more proficient in the use of these tools that, again, they're just having a lot of impact. So I really thank you very much. I hope that, again, that next time this is in person. And stay safe, and thank you everybody in the audience for listening and for your participation. And I hope you see a spike in users from Latin America after this presentation.
I will be looking for it.
Yeah.
Thank you so much, Gabriela, for having me. Thank you everybody who was listening, and anybody can contact me through emails with any questions if you would like later. Thank you.
Thank you. Bye-bye. Have a nice evening. Bye.